I'm here with Dr. Kah Fei to discuss a little bit about target translation. Dr. Kah Fei, would you briefly introduce yourself?
Hello everyone, my name is Kah Fei. I'm a pharmacist and cancer pharmacologist by training, and currently I'm heading the antibody biology group in EDDC.
So Dr. Kah Fei, people talk about target discovery in the drug discovery space. What part of that space constitutes biologics?
Firstly, due to the large size and surface, biologics can target biological mechanism that is intractable to small molecule drugs. For example, we have antibodies that can block soluble factors such as VEGF, vascular epithelial growth factors, and also TNF, tumor aggressive factors. We also have siRNA that can block transcription factor functions, therapeutics proteins that are very effective as agonists, and we also have both therapeutics proteins as well as antibodies that are ideal for targeting protein-protein interactions. Biologics can also offer great specificities without significant and unpredictable off-target toxicities and longer durations of actions. The comparatively high approval success rate for biologics from first in humans to launch is one of the reasons for the growing interest for the development of biologics therapeutics in both the pharmaceutical as well as the biotech.
Would you say that this increased interest in biologics is a recent phenomenon, or has it been for a few decades? Or
I think it's been at least a decade from now when it comes to the growing interest in both the pharmaceutical as well as the biotechs in investing in the biologics research.
If I understand correctly, as opposed to small molecules, biologics are big molecules. So what cellular molecules are considered biologics, and why are they good as therapeutics?
That's another great question. So 60% of the target space can be targeted by biologics, and that consists mainly of secreted proteins, soluble ligands, and extracellular membrane proteins. As you know, secreted targets and soluble ligands regulate many important processes, and indeed they're the first class targets for biologics. Cytokines and growth factors are the predominant types of targeted secreted proteins. For example, TNF has been one of the most targeted ligands. There are at least 5 approved anti-TNF biologics. Humira, also known as adalimumab, is one of the best-selling drugs for the treatments of rheumatoid arthritis. Another great example that is heavily targeted is growth factors, vascular epithelial growth factors, VEGF, as a drug target focused on its role in tumor angiogenesis. Anti-VEGF antibody bevacizumab became the first drug against these targets to reach the market when it was approved in 2004 for the treatments of colorectal cancers in combinations with chemotherapies. It is now approved for the treatments of multiple cancers.
Wow, that's a landmark approval for the first time.
And it's still one of the best-selling drugs.
Dr. Kah Fei, within the field of antibody therapeutics, there's so many targets that people can study. So what qualifies some targets as better than others?
Just like any other targets, some general consideration is also applicable to antibody. First, it is important to build and establish the strong evidence for proof of mechanisms into the discovery paradigm. This includes demonstrating the targets can modulate the disease in a specific manner. Second is to eliminate the targets that have a mechanism-based toxicities. And this risk can be interrogated during discovery using molecular tools such as gene knock-in or gene knockout, or to examine the target expressions in the disease versus the healthy settings, which we can leverage on the publicly available database such as TCGA, Atlas. Alternatively is to use the 2 antibodies from the commercial source to examine the target's protein expression in either the cell lines or primary tissue samples. For example, you may want to perform flow cytometry, analysis, or histology studies to confirm the surface expressions for surface targets. Another consideration is whether there is any tool that can be used to identify biomarkers that signal the correct dosing, and whether the specific molecular targets has been hit, and the information will be very valuable even at the discovery stage. Lastly, it is important to identify the appropriate animal models for efficacy testing in preclinical studies. One of the biggest challenge is the lack of fidelities with most disease models, and a lot of times this model actually fail to recapitulate the actual disease microenvironments, hence does not actually accurately reflect how disease naturally evolve in the patient. Recently, we have begun to see a lot of advancements in humanized models or ex vivo organoids model making their way, and going forward, I believe these models will provide us with a better transferability in terms of target efficacy and safety.
Right. A quick follow-up on the fidelity of animal models. Do you mean fidelity from the genesis of a disease like cancer, let's say? The progression of that cancer as it happens in an animal model is different than how it happens in the humans?
That's
and
correct.
That's what you're calling fidelity?
That's correct. Okay. Or even, some of the robustness of the animal model is actually, lacking or much poorer. And it's a lot of time you may not be able to reproduce, the same results in the same disease setting as well.
Yeah.
Yep.
It makes sense for ex vivo models as the new.
That is definitely, that's definitely, I would say, the ex vivo models. For example, organoids, that is much more, higher throughputs, and that it can go through lots more manipulations, and how that actually reflect what's happening, in the human's disease settings as well.
So let's say now you have a target that you want to go after for antibody therapeutic development. So what are some of the, in your opinion, key factors that play a critical role in the development?
So as a general rule, identify your targets, validate your targets with greater emphasis on how reproducible the data is, followed by drug discovery campaign. So just like any other drug discovery campaign, there is a need for us to develop a robust flowchart to distinguish the true hits from the non-specific ones. Typically, the campaign consists of iterative cycle of screening, optimization, and selections. Screening is to identify not only the antibody binders, but also functional antibodies that show cross-species reactivities. Second is the optimizations to improve the affinities, potency, cross-species reactivities, and also developabilities. And of course, we wanted to reduce the immunogenicities by antibody humanizations if this is not a human antibody. Third is the selections, is to select potent antibody to deliver the right pharmacology and full developabilities, and this becomes the critical elements here. An ideal preclinical development candidates will fulfill all the target antibody profiles that is differentiated from competitors. That's something you can patent, good developability potential with good in vivo PK's properties. In parallel to the PDC selections process, lead indication selections, strategies for patient selections, and also biomarkers are further validated and strengthened to support the translational medicines plan. However, just like any other drug discovery process, there are several common challenges that we encounter during the hits to the PDC selection stage, which can be classified into several categories. First, usually we can find suitable antibody, but that may require higher dosing than desired based on limited potency. Second is we could find good antibody, but developments may be difficult because of limited cross-species reactivities. And third, antibody with great pharmacology, but we have serious concerns and worries about the developability and the in vivo utilities. So by understanding how all these molecules' properties behave and identifying the potential issue in each categories, we'll be able to devise a mitigation strategies, including combining the strength of each molecules and optimize them into one single all-rounders molecules, or we can consider using a surrogate molecule for pharmacology and diagnostics applications. By having these informations, this will actually allow us to define whether this fit into the first-in-class or the best-in-class strategies, and how we can actually bring the molecules into the clinical proof of concepts as soon as possible.
Right. Wow. Thank you. Thank you for that very deep dive into what's involved in developing an antibody therapeutic. I'm sure you'll get a lot more questions from the interested parties who want to engage with you for further target discovery and development. And I thank you for your time again.
Thanks, Shiv, for your questions.
Yeah.
Thank you.